what I want you to keep in mind is this question: can you get there from here? This is a huge question for major transitions in the evolutionary world. Can you get there from here? With respect to large information-bearing sequences in our own experience of human language and computer code, we realize it's very hard, if not impossible, to get there from here by a series of random changes. This talk that I'm about to give may have some technical material that's a little bit challenging. And I wasn't sure that this was the right place to give this talk, except that, number one, it's so timely to discussions that are going on in biology right now. And number two, it has the most amazing payoff for the science-faith discussion I couldn't resist. So I'm going to give it a shot, and I'm going to give you guys, I'm going to stop myself periodically to check in with you all to make sure I haven't lost people. And I'm also thinking of a whole bunch of ways to try to explain this as clearly as I can. One of my colleagues and friends here is Mark Monty, who did a wonderful technical presentation seminar for us in the fall with some of our scientists. And he told us that we have to guard against the curse of knowledge and talking as if everyone already understands what we're saying. So I'm going to try not to do that. In fact, I'm going to start the talk before I even begin getting into it with a question and an illustration. Imagine that you are a computer programmer and you have just written a wonderful block of code for a new app, a new computer application. Now, what would happen if some of those digital bit strings, those zeros and ones that are part of your code, were, for whatever reason, randomly changed? Could you, through a series of random changes to those digital bit strings, ever hope to get a new app? Okay, get the problem? I mean, all the computer scientist people are rolling their eyes at the question, right? You're going to degrade the function that you have in the first place long before you ever get to a new app or a new program or a new operating system or anything useful. Now, there's a reason for that, a mathematical reason that we'll get into later, but I just want you to keep that in mind as we begin. In fact, here's a linguistic example of the same thing using English text. Imagine you start with the phrase, "time and tide wait for no man," and you want to evolve by random mutations or changes a line of Shakespeare, "Methinks it's like a weasel." What's going to happen? Well, the first few changes, you might still recognize the original phrase that you had, but eventually you're going to drop into a zone of the functionless abyss. You're not going to be able to make head nor tail out of the sequence. Now, in the evolutionary process, the idea is that you can change things gradually, provided you maintain function all the way along the way, step by step, because natural selection, the Darwinian mechanism, selects for functional advantage. If there's an advantage to a variation that arises naturally, it will be preserved and passed on. But if at any point you lose function, those variations are not going to be preserved and passed on, and the evolutionary process will terminate. And so what I want you to keep in mind is this question: can you get there from here? This is a huge question for major transitions in the evolutionary world. Can you get there from here? With respect to large information-bearing sequences in our own experience of human language and computer code, we realize it's very hard, if not impossible, to get there from here by a series of random changes. Okay, how are we doing so far? This is the key idea of the talk. Key idea. We're going to come back to it in different ways. Okay, now quick review of the key argument for intelligent design, or rather a key argument. I heard the talks this morning. There are so many wonderful arguments, but this is the one that I've worked on. And that is that when we see the abrupt appearance of new form, whether it's the first life or new forms of life in the history of life as preserved in the fossil record, we are also, this is a depiction of an event in the history of life known as the Cambrian explosion, where we get most of the major groups of animals arising very abruptly in the fossil record. It's mysterious from a Darwinian point of view because it does not, the pattern in the fossil record does not match the tree-like pattern that Darwin expected to find. The gold dots represent the abrupt appearance of all the new forms of life. The blue dots in this diagram represent the transitional intermediates or ancestral precursor forms that you would expect to find in the fossil record if they had arisen, if the new animals had arisen in a gradual, step-by-step fashion. And you see that this pattern, the tree-like pattern, doesn't really match the abrupt appearance pattern. Gold dots with nothing filling in the blue dots. We don't find anything in the fossil record that look like ancestors to the first arthropods, trilobites, that is, fish, jellyfish, etc. But that's not the deepest problem. As I argued in Darwin's Doubt, we have the mystery of the missing fossils, but we also have the mystery of how to build an animal. And the problem is that we now know in 21st century biology, in fact, we've known since the 1950s and 60s, that if you want to build a new form of animal life, you need to have a lot of new information. Biological form requires information. And so let me give you another example from the computer world. If you want to give your computer a new function, what do you have to give it? Code, information, digital bit strings, okay? Very specific code. And the same thing turns out to be true in life, and I can illustrate why that's true very quickly. If you have, for example, the first fishes arising in the Cambrian, and you look at all the different types of cells that are in a fish, and then you look at the animals that are in the Precambrian period, you find that there's a lot of types of cells that were not present in any of the Precambrian animals. A sponge, for example, had between 6 and 10 cell types. A Cambrian fish had 60 or more. Now, I'm not arguing that the Darwinists think that fish evolved directly from Precambrian sponges. The point is rather that nothing in the Precambrian period had all of the cell types that come online when you get these new animal forms. So you've got to account for the new cell types that make new types of organisms. But new cell types require new proteins, and new proteins are built from digital information in DNA, from code. So for example, if you have a Cambrian fish, it's going to have a gut. A gut will require enzymes, digestive enzymes, to process food. Well, those are forms of proteins, and proteins are constructed as the result of the instructions that are in the DNA molecule. So, the problem of the origin of new form got a lot more acute beginning in the 1950s and 60s as biologists were discovering the importance, the primacy of information to biological organisms. New form requires new information. Every time you get a new form, you need you get a new body plan, which is a new form of life. You need new organs, new tissues. New tissues require new cell types. New cell types require new proteins, and the proteins are built from the instructions in the DNA molecule. So at the very least, you need a lot of new genetic information. We learned about where that information is stored from Watson and Crick. They pointed out, or Crick pointed out in his famous sequence hypothesis, that the constituents in the DNA molecule, the constituents parts called the nucleotide bases, are functioning like alphabetic characters in a written text or like the zeros and ones in a section of software. Thus, for example, Bill Gates has said that DNA is like a software program, but much more complex than any we've ever created. So we've got information in life, and the information is necessary to build the parts that we see when you get a new form of life arising. You need information to build new form. And the question is, where does that come from? And also, let's just do a little review on what it builds. Hold on, I left a visual aid in my bag. I told you I was going to try to make this as simple as possible. Okay, I've been mentioning proteins. How many have some kind of familiarity, not just with protein as what you eat in meat, but what proteins do in biological systems? Okay, I'll give you a quick little rundown. Proteins perform all the important jobs in the cell. They catalyze important reactions. Those are called enzyme proteins, and they catalyze them at incredibly rapid rates. They also build the structural parts for molecular machines. Sorry, pay no attention to all these amino acids dropping on the floor. How many have seen pictures of the Michael Behe's stuff, the bacterial flagellar motor and those things? Okay, the bacterial flagellum is built from protein parts, the rotor, the stator, all those parts. Okay, so that's another thing that proteins do. A third thing that proteins do is that they actually process the information on the DNA. Okay, so here I have a chain, and the proteins are made of amino acids, and I've represented the amino acids with these little tiny beads of different colors and shapes. And that's because amino acids, the constituent parts of DNA, do link together in the way I've depicted here, and they often have different chemical structures. And those amino acids, when they're linked together in particular ways, will exert forces on each other that will cause the whole chain to fold into some very distinctive shape. Now, there are many ways it could fold or not fold, but if the chains fold in very specific shapes, then they're able to do jobs in the cell. They're able to catalyze reactions and build structural parts, process information. So the proteins are built from the instructions in the DNA. The DNA sends instructions to a manufacturing center in the cell, which puts together the proteins in a step-by-step way, one amino acid at a time. If they're arranged properly, they fold into three-dimensional shapes that will do specific jobs. So this is a picture of the chemistry of amino acid sequencing, and then here's a picture of an enzyme that's formed from the proper sequencing. It's got a particular shape. In this case, it is actually a digestive enzyme, and it's breaking apart a two-part sugar and producing a glucose and a fructose because of the hand-in-glove fit, because of the hand-in-glove fit between that enzyme protein and the molecule that it's going to act on. Okay? And proteins have this feature of three-dimensional specificity. If you get the amino acids in the right sequence, the proteins will fold into the right shapes, and then they can do very specific jobs. Just like a toolbox has a wrench, a hammer, a saw, each one of those tools can do specific jobs because of their specific shapes, their specific forms. So proteins are super critical, okay? So here's three things we need to know about proteins. First, that specific shapes perform functions, or that is to say, functions can be performed because the proteins have specific shapes, that folded chains make those specific shapes, and that precise amino acid sequencing is responsible for the chains adopting the right shapes. If the sequences are wrong, if the arrangement of the amino acids is off, it'd be like having a misspelled word. You couldn't make sense of it, okay? A badly misspelled word. All right? So you see that the shape of the protein in the end is the thing that's really important. And so there's something about proteins that there's a way of describing those shapes. The protein scientists call them folds. They're the very distinctive shapes that allow very distinctive functions to be performed, okay? And all of that information for building the protein fold that allows the functions to be performed ultimately comes from the DNA molecule. How many are familiar with CAD/CAM? Okay, so engineers. Or what about digital printers? Do people know about digital? Far more people know about digital printers than CAD/CAM. So you know that today we have technology that allows us to write digital code and produce three-dimensional objects. At Boeing, they use digital code to direct the construction of the airplane wing. So the engineer writes the code, goes down a wire, it's translated into another machine code, and then that code is used to direct a manufacturing apparatus to, say, place the rivets on the airplane wing in accord with the specifications of the engineer. Now we have also these digital printers. I was so confused about these when I heard about it, that how do you build a doorknob but with a printer? But it's kind of a metaphor, printer. So we now take digital code. You can build a garage door or a doorknob or a piece of pottery. So digital information constructing three-dimensional objects. That's what's going on inside cells. Okay, we've just invented this. We're just catching up with what's been going on in biology since the origin of life. It's crazy, okay? But the key thing now I want you to keep in mind is the protein fold, because that's the outcome of this manufacturing process. All right, how am I doing so far? Okay, this is really the scientific background you need to understand the argument I'm going to make, all right? Okay, so importance, and here's the important thing about the folds. The unique three-dimensional shapes of the folds make new and unique functions possible. And here, back to this little picture. This is just a picture of 4 of these distinct shapes. There's several thousand folds that we know about, and they're very important in biology. And they're important because their unique three-dimensional shapes make possible new and unique functions, okay? Secondly, folds represent the smallest unit of structural or functional innovation in the history of life. They are, in a sense, the fundamental forms. All other forms are built, all other forms require first the building of protein forms. So if the evolutionary process cannot explain the origin of the protein fold, it's in trouble. You've got to be able to explain where those folds come from. And, I came up with a new trendy abbreviation. I've learned about this IMHO, in my humble opinion. I have a new one. IOW. In other words, okay. In other words, all structural innovations in the history of life depend upon protein folds. If you can't build protein folds, you can't build new animal forms. You can't build new cell types. Okay. So back to our Cambrian explosion as an illustration of one of the big events in the history of life where there's a great amount of morphological innovation, that is innovation of form. We see you've got to have a lot of information. Get from a single-celled organism to an arthropod, you jump from several 100,000 digital characters, base pairs, to 120 million. Big jump in information at the Cambrian explosion, just for that one animal. Okay? But what do we know about information? This is the ID argument. Information comes from a mind. There's an early pioneer in the application of the information sciences to molecular biology, and he in an offhand statement at one point said, the creation of new information is habitually associated with conscious activity. So when we see these big jumps in information, we suspect a mind played a role. Again, Bill Gates. Human DNA is like a computer program, but much more complicated, much more complex than any we've ever devised or created. Where do computer programs come from? They come from minds, right? So in my books, I've argued that whenever we see information and we trace it back to its source, whether we're talking about a computer program or a paragraph in a book or a hieroglyphic inscription or information embedded in a radio signal, or I could go on, we always come to a mind, not an undirected material process. So the discovery of information first at the foundation of life, and then the big jumps of information we see throughout the history of life suggests the activity of a designing intelligence in the origin and history of life on Earth. That's the argument. That's a sketch of the argument. Now, to make that argument stick, you've got to also show that there isn't another cause that's sufficient to produce these big jumps in information. And that's what I want to talk about now, because the evolutionary process has been proposed as a way of generating information. In particular, it's been argued that random mutations and natural selection themselves generate information. They generate new genetic information for building new proteins and new protein folds, and therefore, eventually, new forms of life. And so the question I want to ask is, well, can that mechanism accomplish all that? Can random mutation and natural selection produce the genetic information necessary to generate these crucial structures called protein folds? And one of the really exciting things about focusing on the protein folds is that's a place where the question of the creative power of the mutation selection mechanism can really be adjudicated. It's a tractable problem. There's a way of analyzing that question, the one I've just posed, through experimental evidence that will help us determine whether the evolutionary process can generate those new important structures or not. If you get to a higher level in biology, you can get a lot of hand-waving on both sides of the argument. This is a place where there is experimental evidence that makes the question tractable. It allows us to assess whether the mutation selection mechanism can do the job. Now, again, we've already hit this intuition a couple times, but let's come back to it. The intuition that many people have, because this is borne out in our own world of English text or computer code, is that random changes to sequences of specified information, functional information, will invariably degrade the information present in those sequences. And we see that in computer code very readily, because it's really fussy. Now, there was a scientist, there was a famous conference back in the 1960s at MIT, convened by MIT scientists called the Wistar Institute Conference, where some of these concerns about the creative power of the Darwinian mechanism and its ability to generate new information were first laid out. The mathematicians and the engineers and the physicists at that conference were skeptical that mutation and selection could generate new information. And one of the reasons, this is a quote from one of those scientists, Murray Eden, a professor of actually engineering, computer engineering there, he said, "No current existing formal language can tolerate random changes in the symbol sequences which express its sentences. Meaning is almost inevitably destroyed." Well, that's our same intuition again, right? Degrade the random changes, you're going to degrade the meaning. But it turns out there's a mathematical reason for that. And I can illustrate it first with English text. If you have, say, a 12-letter sequence in the English language, and you ask yourself, well, how many 12-letter sequences are there that are meaningful in comparison to all the different ways you could arrange the letters on the Scrabble board from the alphabet that are gibberish? And at first, it's a hard thing to get your mind around, but it turns out that for every meaningful word 12 letters long in English, there are 100 trillion other ways of arranging the 26 letters of the alphabet that will give you a non-meaningful word, that will give you gibberish. Which is to say that the meaningful sequences are rare among all the possible combinations of the 26 letters of the alphabet of that same length. Does that make sense? Okay. So now there's a concept that scientists use to talk about the size of all those other combinations, or how many other combinations there could be. And they call that sequence space. You think, okay, there's 12 letters, and how many possible ways are there arranging those 12 letters? There's 12 spaces. How many different ways are there of arranging the 26 letters that are 12 spaces long? And whatever the answer is, we call sequence space. Or sometimes it's called combinatorial sequence space. It's just saying, how many combinations could you make from the, and that's where it gets really crazy, because if you think, well, if there's 26 letters, it's 26 times 26 times 26 times 26 times, so it's 26 to the, that's a big exponential number, right? Okay, and that's why it comes out to 100 trillion. Is something like that going on with genes and proteins? With the genetic text, we got English letters, lots of different ways of combining them. Well, could it be the same with the A's, C's, G's, and T's in DNA, or the amino acids and proteins? That there are a lot more ways of arranging those characters or those elements, those subunits, than there are ways of getting functional versions of those combinations? Because if so, that would be very interesting. And it turns out that that's something that has been addressed experimentally by one of our now key colleagues, Douglas Axe. He spent 14 years at Cambridge University addressing this question: how common or rare are functional sequences? He was focusing on proteins, but remember, protein sequences are derivative of DNA sequences, so he's asking the question about DNA or proteins. How common or rare are the functional arrangements, the sequences of amino acids, among all the possible combinations that you could make with the amino acids? Is it kind of like English language, where there's rare little islands of function amid a vast sea of gibberish? And it turns out that, yes, the DNA sequences and the protein sequences are very much like the case with English language. In fact, it's more acute. Here's a way to think of it. For every one of these, this is his important question, for any one of these folded structures that will perform functions, how many are there of these? Where these are, like the old pizza in the Saturday Night Live skit, where they just flop and don't do anything. Okay, non-functional gibberish sequences. How many of these are there how many of these are there for folded structures that will do a job? And he used a sophisticated method of sampling in molecular biology called site mutagenesis, and this was his answer, the answer he derived, that for every functional protein fold, or for every functional protein, there's 10 to the 77 ways of arranging the amino acids of the same length, in the same length, that will produce a nothing burger, something that won't do a job. Okay? Now, why would that be a problem for the evolutionary process? Okay? Well, let me illustrate. Evolutionary process depends on, first, a random set of mutations. Sometimes people think natural selection is creative. No, natural selection acts after the fact of random mutation doing its job. Natural selection selects for functional advantage among naturally arising variations or mutations. So it's essentially relying in the first place on a random, unguided search for a functional outcome. If the functional outcomes are super rare and hard to find, you could be looking till the cows come home and never find something that natural selection could select and preserve and pass on. See the problem? Natural selection acts after the fact of a successful random search. So what we need to assess is how hard would it be to find a functional protein, or worse yet, protein fold, given axis numbers. All right? How are we doing? Still everyone with us? Okay. See, these numbers are prohibitive. They're a little scary if you're a Darwinist, right? So here's a way of illustrating the problem. Imagine that I'm a bike thief, and I like to rip off people who come to science and faith conferences at nice churches in Dallas. And there's some nice bicycles out there that have been locked up. And I encounter a 4-dial bike lock. Is it more likely that I will succeed? And I don't know the combination, so I'm just going to try to crack it at random. Is it more likely that I'll crack the code, that I'll find the combination at random, or that I will not find the combination at random? Okay, you all fell into my trap, all right? If I have limited time, yes, it's more likely that I will not find it, right? But if I have unlimited time, or I can keep after, if I'm a particularly devoted or determined thief, I've made these calculations. It turns out that if you turned one dial every 10 seconds randomly to sample all the possibilities, there are 10,000 possibilities on a 4-dial lock, 10 times 10 times 10 times 10, that's 10,000. And there's only one that will open it. But if I try one every 10 seconds for 15 hours, I'll get to the point where I've tried 5,001 combinations, and then it's slightly more likely that I'll succeed than I won't. Okay, so I've sampled more than half, and it becomes more likely. So when we're assessing the plausibility of a random search, we need to know two things. We need to know first, how vast is the space, the sequence space of all the things I got to search? How rare, and then how rare is the thing I'm looking for, but also how much time do I have to look? Actually, that's three things. How big is the space? How rare is the thing I'm looking for? And how much time do I have to look? All right? So if it's just a 4-dial lock and I'm really determined, I could probably crack it. It's more likely than not that I'd crack it if I stick to it for 15 hours. Okay? Sampling. But what if I encounter a lock like this? A 10-dial lock on the bicycle out in the parking lot. Well, now it turns out if the thief is numerate, able to make calculations, he'll realize this is a waste of time, because if the thief has 100 years and does nothing his whole life but turn dials every 10 seconds, no potty breaks, no meals, no coming to science and faith conferences, he will only be able to sample 3% of all the combinations. In which case, it will be much more likely that the thief will fail than it is that the thief will succeed. And if you're a betting person, you're going to bet on the failure, not the success. That is to say, it's a better hypothesis to say that the chance hypothesis is wrong. All right? So what's the case with DNA and proteins and axis numbers? Are the genes and proteins more like the 4-dial lock where might be enough time to crack it? Or is it more like the 10-dial lock where you don't have enough what are called probabilistic resources to have a reasonable chance of a random search succeeding? Okay? Well, the number's very big. It's 10 to the 77. That's a big lock. I've got one here, 77 dials, okay? Searching. But we do need to know how many opportunities there are to search. How long can you keep after it? Well, if we take the standard date for the origin of life on Earth at 3.85 billion years, or even go back to 4.5 billion years for the origin of the Earth, it turns out there's only been 10 to the 40th organisms that have ever lived in the history of the planet. So, and every time there's a new organism arising, every time there's a cell division, then you could have a new mutation that would be passed on to the next generation. So you've got 10 to the 40th possible trials. That's a big number, but it's small in relationship to 10 to the 77. If you have 10 to the 40th trials, but the size of the space you're searching is 1 in 10 to the 77, that means you're only going to search 1 10 trillion trillionth of the total possibilities in the known history of life on the planet. So then it becomes overwhelmingly more likely, overwhelming and quantifiably more likely that you'll fail, a random search will fail, than it is that such a search will succeed. In which case, it's also more likely that the chance hypothesis that that's how it happened is false rather than true. Okay, if it's a bad hypothesis. If you're a betting person, you're not going to bet on that hypothesis because it's overwhelmingly more likely that it's not going to work. All right? And since natural selection can't help you until you find that functional structure, that protein, good luck. I mean, the bottom line is it's not an adequate mechanism to produce the new information that's required to generate a new protein. And I'm going to say also, or a protein fold. It turns out that the protein folds are even more rare than the sequences around a single fold that acts sampled. And that's a technical point I'm not going to go into. In other words, 1 in 10 to the 77 is a best case scenario number for the Darwinists. If you want to get a fold, it's going to be even more difficult. Now, there's something else that supports all this, and that is not just that the proteins in the folds are rare in sequence space, again, all the combinations of amino acids of a given length that could be, it's also that they're highly isolated from one another. There's an Israeli scientist, a protein scientist named Dan Tawfik. Alas, he tragically died just a few years ago, but he had, at the time of his death and before, he was doing cutting-edge experiments on protein folds and seeing what happens to them, the key structures that allow functions to take place, those fundamental units of biological innovation. He's doing experiments on them, and he's randomly mutating them. And he finds as he randomly mutates the folds, they start with these beautiful three-dimensional structures, and between 3 and 15 mutations usually cause the thing to go limp, like the pizza in the Saturday Night Live sketch, okay? And so this suggests that, well, because folds typically are different from one another by about 65% of their sequences, and this number is, less than 5 or 10% depending on the protein he's working with, that's not enough changes to get from here to there. In other words, his results suggest you're falling into a functionless abyss before you ever get to a new protein fold. You can't get there from here. You'll destroy the stable structure that makes function possible before you get to a new stable structure that's functional and selectable. Because you've got to maintain function at each step along the way for the Darwinian process to work. Okay? And Tawfik studied the same protein that Doug Axe studied, but also some others, suggesting that these results are generalizable. And though he was no friend of intelligent design at all, I can have some backstory on that, he ends up concluding that the origin of protein folds is akin to a miracle. You can't get there from here. We have no explanation for the origin of folds. Now, he's not saying you can't mutate proteins and have them optimized. You might have a fold, you may start with a fold, and you can, if you maintain that fold, you can optimize the function of the protein. You might even get some slightly different functions being expressed by that fold, but if you want to get fundamentally new functions expressed by fundamentally new folds, you're going to destroy, as the mutations accumulate, You're going to destroy the function of the original fold. Well, it's worse than that. You're going to destroy the thermodynamic and structural stability of the fold long before you'll get a new stable protein fold. You're dropping into an abyss, functionless abyss, okay? Now, and so we're back to our original illustration. You've got a zone of function, you start to change things, you drop into an abyss before you ever get to the new zone of function. All right, now, I actually discussed all this on Joe Rogan. And there's a little clip of me, about 4 minutes long. Joe Rogan asked me, do you believe in evolution? And it's always a trick question because, of course, you could believe in microevolution and you believe in change over time, you might believe in common descent. So I gave him but I said, I'm profoundly skeptical about the creative power of the mutation selection mechanism. And then I explained why. And what I've just explained to you, I explained to him in 4 minutes. Okay? Now, I don't know if I can get this to make noise and play. Let's see what we can do. There we go. I think we're hearing sound.
Skeptical about what's called universal common descent, the idea that all living forms.
See if you can follow this now, given what I've just said.
Common ancestor. I'm also skeptical about the creative power of the mutation selection mechanism, which, as it happens, so are many leading evolutionary biologists today. So, modification, yes; innovation, no.
So modification over massive amounts of time. Don't you think that would eventually lead to new groups? Because a lot of new groups have similar origins, or at least origins from one ancestor.
Well,
time
was always the hero of the plot. But let me just run a couple of arguments by you, and let's see what you think. Okay, and I developed these in a lot of detail in my book Darwin's Doubt. If we now know, thanks to the genetic revolution, the molecular biological revolution, that if you want to build a new form of life, you have to have new code, because all new forms of life depend upon new anatomical, a fundamentally new type of animal, for example. So you need new anatomical structures but the new anatomical structures require new cell types. New types. So if you got animals that first come online and they have a digestive system, they have a gut. Well, you got to have enzymes that can service a gut, that can process food. So enzymes are types of proteins. Proteins are built from the informational code in DNA. So anytime you want to get it's just like in the computer world. If you want to give your computer a new function, you've got to provide new code. So we have these long digital bit strings. A's, C's, G's, and T's, not zeros and ones, but A's, C's, G's, and T's in a digital string, and we call that a gene. And if you have a section of DNA for building a protein, that's great, it all works. No, but if you want to build a fundamentally new form of life, you got to have new proteins to service the new cell types to build the new anatomical structures. In our computer world, we know that if you start randomly changing the zeros and ones in a section of digital code, you're going to degrade the function of that code long before you come up with a new string for making a new program or operating system. The functional sequences they're highly isolated in what's called sequence space. You can change a few things and still retain function, but after a very few number of changes, you're going to degrade the function, and long before you come up with a new function. Now, the Darwinian mechanism starts with the idea that there are random changes in those digital bit strings, those sequences of A's, C's, G's, and T's. And based on our experience in the computer world, we would expect that random changes are going to, again, degrade those strings long before they're capable of building a new protein. And there's now very compelling experimental evidence that that's true. There's an Israeli molecular biologist, Dan Tofik. Unfortunately, he died fairly recently in a tragic accident. But he was doing mutagenesis experiments on sequences of code for building specific proteins that fold it into stable structures. They're actually called protein folds. And he found that between 3 and 15 mutations was enough to degrade the thermodynamic stability of the protein structure that the gene was making. And once you lose that thermodynamic stability, you have no functional possibilities.
Is there possibly an undiscovered mechanism for protecting against that we're not aware of yet?
Possibly, but there's numerous lines of evidence suggesting that that mutations are within limits. You can modify again, you can optimize an existing protein structure called a fold, but if you allow too many of those mutations, you're going to degrade. And long before you would get a fundamentally new protein structure, another protein fold.
I can't believe I really tried to explain all that on Joe Rogan, but there it is. And I hope that answer makes a little more sense if you saw that based on what we've just been talking about. Now, let me give you the counter argument to all this, because this is where it gets interesting. When I was in grad school, one of my supervisors used to say, beware the sound of one hand clapping. Okay? If there's an argument on one side, there's bound to be an argument on the other. So I want to give you the counter argument to what I've been arguing, because interestingly, it's been advanced by people who have a new view of what's called theistic evolution. Okay? So the biggest people, the people who are most coming to the defense of Darwinism right now, or evolution, are theists who are trying to, in a way, combine evolution with intelligent design. And it's an interesting proposal, so let's have a look. So the question is, are the proteins, yes, we grant that the proteins are very rare in sequence space, but is it really true that they're isolated from one another? Is it possible that there might be a propitious pathway from one to the other, where where you can maintain function at each step along the way, or at least in kind of stepping stones to get from one island of stable, selectable protein structure to another? So yes, they're rare in the totality of all the possible ways there are to arrange amino acids, but could it be that there's a very a very narrow pathway connecting one to another? And this is an idea that's been put forth by two young researchers, one a theologian from Finland named Ropi Kajonen, another a biologist, a New Zealander who's working in England, Zachary Arden. So they ask, well, what if there is a continuous pathway of functional protein sequences connecting one fold to another? And they have a way of depicting that. Now, this is the way you'd depict the idea of isolation. Each one of these plateaus represents a fold. Each one of the spikes represents different variations of the function that the fold performs. But notice in this way of depicting it, you see that the folds are isolated from one another. And biologists have a term for this type of depiction. They call it a fitness landscape. The peaks show fitness, a protein that's doing a job. If you drop below the water level or drop down to the valley, then you're in a non-functional abyss. Okay? Now, what Rope and Zachary have proposed is a fitness landscape that looks more like this, where you have folds, but then you have a little archipelago of islands connecting one fold to another. Could that be the case, they suggest? Or here'd be another way of looking at it, where the land masses represent two protein folds, and instead of being isolated like this picture shows, they propose that maybe there's an isthmus, a land bridge connecting the two. Or just to do one more metaphor to get our minds around this, if each of these rocky masses or islands represent a different fold, they suggest that, well, maybe there could be a kind of archipelago of islands suggesting or connecting from one to the other. So yes, the land poking up above the water, which represents the functional folds, or the folds that are capable of functions. Yes, it's rare in the ocean, but there's this very nice propitious pathway connecting the folds from one to another. Now, if you were to see a pond and you saw stepping stones from one side of the pond to the other that were lined up like this, what would you infer about the alignment of the stepping stones? They had been designed, right? And to their credit, that's exactly what Zachary and Rope think. They're suggesting that there is a designed pathway, a continuous pathway between the folds. And that's a very interesting proposal. But notice what is presupposed in all of this debate. We think rarity points to isolation, but we also think rarity points to the need for intelligent design. A random search will never result in finding the folds. So it's got to be guided, it has to be intelligently designed in some way. So either the folds are independently designed or separately designed, or the pathways are designed. Okay, so in a sense, both sides are now operating within a design framework. But what is the case empirically? Are the folds isolated and therefore evolvable along these pathways or not? What do we know that could help us adjudicate that question? So now we get into some empirical evidence. And it turns out there are problems with this continuous fitness landscape idea where you have the design pathways. And the first problem is that the mutagenesis experiments that have been performed by people like Douglas Axe and Dan Toffick show that structural stability of the fold is typically lost before 10% of the amino acids have been changed, and usually well before that. So very few number of changes will result in the unraveling, okay? But separate folds are characterized by 65% or more differences in sequence. Superfamilies of folds are characterized by 80% or greater differences. So the number of changes that take place before you lose structure are, again, not nearly enough to give you a new structure, a new fold. Okay, and that's just something we know. And that would suggest then that as you do these experiments, you're never going to get a new fold, and that's exactly what these experiments have shown. In fact, mutagenesis experiments show that accumulating mutations do not produce new protein folds. They only destroy existing folds before any new folds arise. And it turns out, to use that phrase again, that there is an underlying physics reason for this. That the fold stabilization depends on a quantity in thermodynamics that's called free energy. And with each mutation, you increase the free energy by a measurable amount, and increases in free energy correlate with losses of structural stability. And so usually it's about for chemists in the audience, it's about 1 kilocal per mole per mutation. So we not only know that empirically accumulating mutations destroy structural stability, we have an underlying physics explanation or thermodynamic explanation for why that happens. So this is really solid science. We know that that you get full destabilization quickly before you would ever get the reformation of a new fold. And we also know that when we look at naturally occurring proteins that are likely to have evolved one from another, that we don't have evidence of new folds arising through that. We have evidence, again, of modification of folds, not new folds arising. One more consideration that counts against this designed pathway idea. They want to say extreme rarity, yes, but maybe not extreme isolation. But mathematically speaking, extreme rarity entails or implies isolation. Let me illustrate. Let's go back to our ocean example. Here's the propitious pathway idea, the stepping stones. Now imagine we want to get from say Los Angeles to Japan. Or let's just talk about these two rocks first, and then we'll talk Los Angeles and Japan. Is there enough exposed land in this picture to make a land bridge? Okay? Do you see that there's some level of rarity, there's some degree of rarity where it doesn't matter how well you arrange stuff, you just don't have enough stuff to get from A to B. Okay? And so if we, I've got a couple different examples of this, but is there enough? Okay, I'm doing Los Angeles to Japan. I couldn't remember which illustration I was going to use. Is there enough land here to make a land bridge from Los Angeles to Japan? No, there's not enough, right? Now, what kind of a ratio do we have when we're talking about 1 over 10 to the 77? Well, it turns out 1 over 10 to the 77 is equivalent to, if we do a sphere and we're trying to represent functional proteins as the land and the non-functional combinations as the water, we would need a sphere the size of the visible universe, which, if it's 1 to the 77 ratio, 1 over 10 to the 77 ratio, that would be a sphere the size of the visible universe populated by just 2 dots that combined to equal the surface area of 1 atom. Okay, somebody has done a similar calculation for the ratio 1 over 10 to the 23. That would be like Jupiter with 2 dots equivalent to the surface area of one atom. The numbers we're dealing with, the rarity measures we're dealing with, are so extreme that they entail isolation. There's not enough land, as it were, there's not enough stable proteins in the sequence space to ever make an arrangement that would allow you to continuously get from one fold to another. So these are very compelling considerations that suggest that folds are, as Dan Toffick himself concluded, isolated galaxies, if you like, in the sequence of structure space. There is absolutely no way to connect between them. And of course, we therefore also don't know for any of these enzyme families how they emerged. If they're isolated, they're not evolvable. How are we doing? When Douglas Axe was doing this research in Cambridge, he started to report on the numbers he was getting. And he said, Steve, it's not just that they're rare, he told me. He said, they're extremely isolated in sequence space. I said, what, is it like 2 buttes out on a southwestern desert? And he said, yeah, kind of that's the right image, but it's not like out in the desert in Utah. It's not that they're separated by miles in the desert. It's like they're separated by light years. The isolation is that extreme. All right, I'm a little over time, so I'm gonna cut to the end, and then Daniel's gonna come up, and we're gonna have a little bit of time for Q&A. There's a payoff in this for the science-faith discussion. Notice that now both sides in this discussion, the people who think, everybody's conceding rarity. There's too much evidence of this. It's not just Doug Axe experiments. There's other scientists that have found the same thing. So if proteins are rare, then there's 2 options. Either they're rare and they were designed to be so, that they're information-rich structures that require an intelligence, or they're rare and they evolved one into another through one of these pathways. Now, the theistic evolutionists are saying, yes, they're rare, but the evolution took place on this design pathway. Now, there might be some Darwinians that are out there saying, yeah, they're rare but not isolated, and so evolution can solve the problem. But the problem is the pathways are so rare, too, that you have an argument for design if there are pathways. So I think the debate now is between 2 different approaches to intelligent design. Those who want to combine it with some form of evolution, and others who want to say, look, I think, just if you take the evidence at face value, we're really looking at separately designed fundamental forms that are necessary to biology. So you got 2 basic plausible options. Either you have a pre-engineered, propitiously designed pathway between folds in sequence space, or you have specifically designed individual folds. I think the evidence now supports the idea that the evidence supports isolation, and therefore that there are no pathways through sequence space that could connect protein folds because you lose function on the way. Therefore, and this is the payoff for a science and faith conference, I've been increasingly thinking that what we're studying when we're studying these protein folds, these fundamental structures, they're important to all of biology and upon which all of biological innovation depends. That when we're looking at these folds, we're looking at information-rich, fundamental biological forms that must be, because of their information content, intelligently designed, and because it's not a form of design that morphs one into another. We're actually looking at expressions of individual thoughts in the mind of God. What St. Thomas Aquinas called formal or exemplar causation. Ideas in the mind that result in form in matter. And I think that's what we're seeing. And I'm going to stop there and let Daniel come up, and we'll have a little more discussion about all this. But thank you very much.
Okay, so we're going to do this a little differently than some of the other Q&As, in part because I think Steve is getting it off a little too easy. This conversation about protein rarity and the implications for evolution and intelligent design have been going on for many months now. There have been back and forth conversations, YouTube, interviews, and there's much more to this conversation than what he's been able to cover so far. So
in fact, the Joe Rogan interview has sparked
that's right.
a rather spirited scientific discussion about all this. So
that's right. And one thing in
who knew
particular,
that scientists listen to Joe Rogan?
Apparently they do.
They do.
Right. So one of the things that has come up in response to your Joe Rogan interview is, in reference to this, you mentioned Dan Tawfik's work where he said, 15 or fewer mutations is sufficient to degrade this tertiary functional structure of a protein. So you referenced this research, but I know that in a recent YouTube video with Zachary Arden, that he had some good reasons to suggest that that might not be accurate. And he was talking about the spike protein in SARS-CoV-2.
Yeah, he had a really interesting counterexample. And this is often the way scientists refine their ideas. So he said, okay, interesting argument, Meyer, but there's a protein that we know of, and we're all very familiar with, the SARS-CoV-2 spike protein. There's two different versions of it that are out there, at least two. But when we do sequence comparisons, we see that we have, an Omicron version that's got 37 differences in amino acid sequence compared to the original. So
over the course of only 3 years or something.
Yeah,
like
over
that,
3 years. So obviously proteins can change more than just 15 mutations before they lose their stability, okay? And so this was presented as a counter-argument to not so much what I argue, but what the research that I cited from Tawfik, okay? So you want to know what gives.
What gives.
What gives, okay? So here's what gives. The Arden interpreted me as saying that 15 was a kind of categorical limit in the number of mutations. But rather what I was arguing, what I was using the Tawfik research to do, was to illustrate a principle that's been derived empirically, which is that as mutations accumulate, they inevitably degrade folds before new folds arise. Now, Tawfik, and what we've discovered is that if you do that, if you assess that on a percentage basis, all these site-directed mutagenesis experiments show that it's 10% at the max or less. Now, in the case of the spike protein, the SARS-CoV-2 spike protein, it's a massive protein. It's 1,276 amino acids long, and so 37 amino acid changes is less than 3% of the total length. So on a percentage basis, it doesn't in any way challenge the argument that we made or challenge Tawfik's conclusion. Down in dirty fashion on Rogan, gave the absolute numbers for the proteins he was looking at.
Beta-lactamase.
Exactly, but beta-lactamase has a domain that was being studied that's 153 amino acids long. So in that case, 15 is about 10% at the max. So this is well below that threshold.
And again, the threshold for actually getting some new functional fold is something more like 50, 60%?
Well, exactly right. It's about 65% on average to get the sequence differences between separate folds is about 65% on average. So we're nowhere near that with 3%. And so you'd expect, therefore, on the basis of that consideration, that the SARS-CoV-2 spike protein, the two variants of the spike CoV-2 protein would not exhibit different folds. They would exhibit sequence differences within the same fold structure. And guess what?
Which is indeed true. Because
that's exactly what the paper that Arden cited reported. No new fold has arisen. And this just confirms what Axe and I and others have been arguing, that the mutational process can generate variations that will result in optimized function within the structure of a fold, but not new folds. And we don't know of any counterexamples to that. And the counterexample that Dr. Arden cited does not provide a counterexample. It is a little bit of a gotcha point. Aha, see, there were more than 15 mutations in this particular protein, but it doesn't really refute the main claim, which is that as random mutations accumulate, they will degrade stability before you ever get to a new fold. No new fold was generated, and it was a small percentage of the total. It's exactly what we would expect.
So nowhere in there, though, are you referencing natural selection. So how does the power of natural selection that we're always appealing to, how does that help drive this process?
Well, very good, because in this, to be fair to Dr. Arden, one of the things that he was saying is that in talking about this problem, by citing Tawfik's work, which are experimental results derived from randomly changing an existing, what is called a wild-type protein, a protein as you would find it in the wild, and that when you do random changes, it doesn't take very many random changes to degrade the structure of the protein. But he said that doesn't take into account the role of natural selection in actual living organisms. And that's right, it doesn't. But actually, when you consider a wild-type protein in nature under what's called selection pressure, that consideration actually reinforces our argument and counts against his counter-argument. And here's why. Because what natural selection does, as we've been saying, is that it selects for functional advantage. And it also will act to preserve a stable structure that's performing a function. And so if you start randomly changing something, but you're in the wild, all the random mutations that don't preserve that stable function are going to be weeded out. And then ones that arise that maintain the structure will be preserved. And so therefore you can acquire an indefinite number of of sequence changes if you're under selection pressure and natural selection is preserving the ones that maintain the original stable fold.
And
but if you start...
SARS-CoV-2 is a perfect example, right? Because the spike protein is what's required to infiltrate a cell, replicate, so the moment
and
it
in
doesn't
fact,
bind properly,
right,
you've lost.
Then you've lost, right? Okay. So we're not saying that mutations can't indefinitely accumulate. We're saying that they can't indefinitely accumulate and destroy the existing fold structure. If they're lucky enough, because we know there's lots of different, Axe's work shows there's lots of different variants out there that will maintain the structure. Well, so take 37. Let's say you could get beyond the 10% threshold if it's under selection pressure, because that's what natural selection does. It preserves things that will maintain that structure. It's called purifying selection. So natural selection in this case is actually going to act to conserve the existing structure. And all of this exposes a kind of catch-22. I call it I'll give you a little slide to go with. I call it the evolutionary catch-22. The kind of mutations we need, we don't get. The kind we get, we don't need. The kind we need are mutations that can produce structural change.
Right.
But those are the kinds that are always deleterious, that are always harmful, and that will degrade the existing fold before you get to a new one. The kind we don't need, or the evolutionary process doesn't need if it's to produce new structure, are ones that...
To your idea that these are...
the ones we don't need are ones that alter sequence but don't change structure. Right. So it's not good enough to just say, well, natural selection's at work, therefore it's a magic bullet, it can do anything, because we've got to get a little more fine-grained in our analysis, because what's going on with the natural selection is it's preserving existing form and weeding out anything that comes along that would degrade that. That will allow it to accumulate more mutations, but not mutations that are changing structure and producing new folds. And our argument, again, has been the mutation selection process cannot produce new protein folds. New folds are not evolvable, and since new folds are the fundamental unit of innovation in the history of life, then we've got a serious fundamental problem with the creative power of mutation selection as a mechanism of biological innovation.
Wow. So you're really
that's
natural selection on its head in this case.
It's a real process. We accept that.
Yeah.
But in this case, it's conserving. And what the mutagenesis experiments are showing is they're measuring how many mutations can occur before there's nothing there to select. And the number that are there before there's nothing there to select is not a big enough number to make something to select a new fold. And so it's possible the empirics could have been different. It might have been that the folds were clustered and just a few mutations would give you a new fold. In that case, then there would have been something for natural selection to select. But we know that's not the case, and we are looking at isolation, and therefore the folds are not evolvable, and we have a fundamental problem.
Okay. I think you've defended yourself well, so.
No.
Well, we have just a couple minutes, so I want to ask a couple of the questions that were submitted, which go right along the lines of what you've been describing. This David Zeiger says, scientists like Philip Ball are starting to acknowledge epigenetic information, and they claim that shuffling this information, as well as modular proteins, can result in novel functions. So does this help solve the information gap problem?
Not really, because a lot of sources of what we prefer to call ontogenetic information above the genome, are not mutatable in the way that A's, G's, and T's are along the spine of the DNA. You can readily imagine how, and this was what was so exciting to a lot of the neo-Darwinians after the Watson and Crick revolution, is that they thought, well, this suggests a mechanism for random variation. We could randomly change the A's, G's, and T's. But the problem is that when we realized that the functional sequences were rare in the space of possibilities, it created new problems. So the higher levels of information, whether there be targets, membrane targets, or cytoskeletal arrays, or the sugar code, or whatever it is, these things do not lend themselves to mutation as readily. And so I don't think anyone's really cashed out that proposal with any detail.
So someone says, I think they sum it up well, as I understand it, natural selection favors existing information and removes unfavorable information from populations that doesn't generate some kind of new function. So how can materialists say that this is where new species are coming from?
Well, yeah, I mean, it's a kind of, this is in a sense arguing what I've been arguing, with one caveat. We think that there is a variability. Clearly, there's variability that takes place at the species level, at the genus level, I think possibly even up to the family level. So there's microevolutionary change within limits.
Right.
But when you get to the higher taxonomic groups, I think there's clear evidence of discontinuity. And I think you know, because many different groups within, say, a genus may have the same basic folds as part of their genetic endowment. The argument I'm making is not denying microevolution. That was another thing that Dr. Arden suggested, that you could reduce my argument to absurdity by saying that if what Meyer is arguing about folds is true, then you would never get microevolution. Well, that's not true, because the folds could be part of the genetic endowment of organisms that are within the same species or genus or even family. But I think you get really clear evidence of discontinuity when you get beyond the family level for the higher taxonomic groups like orders and phyla, for example. So, and that's where we see my Cambrian explosion picture was pictures of separate phyla, orders, classes, and phyla, huge discontinuity. And there, I think we really can't enjoin the evolutionary process as an effective explanation.
Okay. Well, we have a lot of other questions that we didn't get to. I would suggest if you have a question that we didn't answer in our discussion, to try to catch Dr. Meyer afterwards. He'll be signing books afterwards. Maybe you can discuss one of those questions. I want to end with just one question to bring it back to home. It's more of a personal question, but I think it's appropriate for this conversation about science and faith and the intersection of the two. This person says, would you say that your training in science has brought you closer to God? And if so, in what ways?
Well, I would say yes, and particularly in the last few months, as Brian Miller and Casey Luskin and Douglas Axe have been diving into this question about the origin of folds. It's breaking new ground for us. And we're really actually grateful to our interlocutors, to Rope and Zachary, because they've posed a challenge that has forced us to look deeper at our own arguments. And in the process, we're discovering new things about folds and proteins that's led me to this realization. I mean, I'm very interested in Thomas Aquinas. He rejected both of the ancient Greek thinkers. He thought that Aristotle's idea of formal causes, Aristotle said that the forms are in the matter. And he said, no, that's going to lead to materialism. Plato said that the forms are in the mind, or in the heavens, not in the mind of God. He didn't believe in the kind of God we do. So, and Aquinas said, no, neither of those things work. Forms or ideas only exist in mind. So Plato's idea doesn't make any sense, but putting all the forms in the matter doesn't tell where the form came from. So he said there were two types of formal causes. There's the form built into matter, but that form in matter must result from an idea in the mind of God, and he called that the exemplar cause. And as I've been studying the importance of these protein folds as a consequence of the DNA, which was my previous fascination, I'm realizing that what we're looking at is a manifestation of an idea, separate ideas that are crucial to life. And it's given me a kind of a sense of awe, somewhat similar to the one I had listening to Bijan Namadi this morning about the planetary system. So yes, I think science, it can create epiphanies, and I've had a few today, so I hope others have as well.
That's a great note to end on. Thank you, Dr. Meyer.
Thank you.